Right now, as your dog lies on the floor or curls up on the couch or follows you from room to room, something is happening inside their skull that most dog owners never think about. Their brain is working, continuously, constantly, without pause, processing smells that arrive in layers of information too complex for human understanding, monitoring the emotional state of every person in the house, mapping sounds from distances that would seem impossible, managing memory, emotion, social awareness, and physical coordination simultaneously, every waking hour, with no days off and no ability to simply disconnect. And the fuel for all of that, the raw material that either supports that extraordinary neurological machinery or slowly degrades it, comes almost entirely from what you put in their bowl. Most dog owners think about what their dog eats in terms of calories, protein, and general health. Very few think about it in terms of brain function. That gap between general nutrition knowledge and what neuroscience has established about canine cognitive performance is where this video lives. These are 5 foods that neuroscience has identified as genuinely supportive of canine brain function. Not supplements marketed for that purpose, not vague claims about natural ingredients, but specific foods with specific mechanisms of action that have been studied, documented, and published in peer-reviewed veterinary and neuroscience research. The first food is blueberries. Blueberries appear on virtually every evidence-based list of brain-supporting foods for dogs, and the research behind them is specific enough to explain exactly why. The mechanism is specific. Blueberries are among the highest sources of anthocyanins, the flavonoids responsible for their deep blue coloring. Research has established that anthocyanins cross the blood-brain barrier in dogs, meaning they reach the brain directly, rather than simply circulating in the bloodstream. Once there, they perform 2 distinct and well-documented functions. The first function is antioxidant activity. The brain is unusually vulnerable to oxidative stress, the process by which unstable molecules called free radicals damage cellular structures because of its high metabolic rate and high oxygen consumption relative to other organs. Oxidative damage to neurons accumulates over time and is one of the primary biological mechanisms behind cognitive aging in dogs. Anthocyanins neutralize free radicals in neural tissue, reducing the rate at which this damage accumulates. The second function is anti-inflammatory. Neuroinflammation, chronic low-level inflammation in brain tissue, is increasingly recognized in veterinary neuroscience as a significant contributor to cognitive decline, behavioral changes, and mood disorders in aging dogs. Anthocyanins have demonstrated measurable anti-inflammatory effects in neural tissue in multiple studies. Research specifically studying dogs has shown that supplementation with blueberries or blueberry extract produces measurable improvements in cognitive task performance, the ability to solve novel problems, maintain attention, and learn new associations, in older dogs whose baseline cognitive function had already begun to decline. The effect was observable within weeks of consistent supplementation. Practically, blueberries are safe for dogs in moderate quantities, require no preparation, and can be offered as treats or mixed into food. A small handful, roughly 10 to 15 berries, several times per week is consistent with the quantities used in research protocols. The second food is fatty fish, specifically salmon, sardines, and mackerel. DHA, docosahexaenoic acid, found in fatty fish is perhaps the most thoroughly researched nutritional component in brain health. It is not merely a beneficial addition to a healthy brain. It is a structural component of brain tissue itself. Approximately 60% of the brain's dry weight is fat. DHA constitutes a significant proportion of the fatty acids in neuronal cell membranes, the outer layer of every brain cell, where it plays a direct role in membrane fluidity, synaptic function, and the efficiency of neurotransmitter signaling. A brain with adequate DHA operates with better signal transmission between neurons, faster processing speed, and more efficient communication between brain regions than a brain that is DHA deficient. Research in veterinary neuroscience has documented that DHA supplementation in dogs produces measurable improvements in learning ability, memory retention, and problem-solving performance. Studies of puppy development have shown that adequate DHA during the first months of life is essential for proper neural architecture, the physical wiring of the developing brain, and that deficiency during this period produces lasting cognitive impairments that are difficult or impossible to fully reverse later. In adult and aging dogs, DHA supplementation has been shown to slow the progression of cognitive dysfunction syndrome, the canine equivalent of dementia, and to improve performance on cognitive assessment tasks in dogs already showing early signs of decline. Salmon is the most accessible source for most dog owners. Cooked, boneless salmon offered once or twice a week provides a substantial DHA contribution. Sardines packed in water, not oil, not brine, are also an excellent and inexpensive option, and their small size means the bones are fully soft and safe. Both can be mixed directly into regular food or offered alongside it. The third food is eggs. Eggs contain choline, a compound most dog owners have never heard of, and that most dog food formulations do not provide in optimal quantities. Choline is a precursor to acetylcholine, one of the most important neurotransmitters in the brain, and it plays a direct and well-understood role in memory formation, attention, and the maintenance of neural structure. Research in both human and veterinary neuroscience has established that choline is essential for the synthesis of phosphatidylcholine, which is a primary component of neuronal cell membranes. Adequate choline supports the structural integrity of brain cells. It also directly influences the production of acetylcholine, the neurotransmitter most closely associated with memory consolidation and cognitive performance. Studies of aging dogs have found that choline supplementation produces measurable improvements in memory and learning performance, and that dogs with higher dietary choline intake show slower rates of cognitive decline over time. Research has also documented that choline deficiency in dogs produces specific and observable cognitive impairments, reduced ability to form new memories, impaired attention, and increased confusion that improve with supplementation. One whole egg, cooked and offered 2 to 3 times per week, provides a meaningful choline contribution alongside high-quality protein and additional brain-supporting nutrients, including B vitamins and vitamin D. The egg should be cooked rather than raw to avoid biotin binding issues from raw egg white that can occur with frequent feeding. The fourth food is pumpkin, specifically plain cooked or canned pumpkin, not pumpkin pie filling. The connection between pumpkin and brain function runs through the gut-brain axis, the bidirectional communication system between the gastrointestinal tract and the brain, through which the state of the gut microbiome directly influences brain chemistry, mood, and cognitive function. Research has established that approximately 90% of the body's serotonin, the neurotransmitter most associated with mood regulation, emotional stability, and a baseline sense of well-being, is produced not in the brain, but in the gastrointestinal tract. The composition of the gut microbiome directly influences how much serotonin is produced, how it is regulated, and how it reaches the brain through vagal nerve signaling and the bloodstream. Pumpkin is one of the richest plant sources of soluble fiber available for dogs, and it has a well-documented and specific effect on the canine gut microbiome. The fiber in pumpkin serves as a prebiotic, food for the beneficial bacterial populations in the gut, supporting the growth of microbiome communities that produce serotonin precursors and other neuroactive compounds. Veterinary studies have shown that regular pumpkin supplementation in dogs produces measurable improvements in gut microbiome diversity and stability, which corresponds to more stable mood, reduced anxiety behavior, and better cognitive function. 2 to 4 tablespoons of plain cooked or canned pumpkin mixed into regular food several times per week is sufficient to produce meaningful gut microbiome support. It is also exceptionally well tolerated, with virtually no known adverse effects in dogs. The fifth food is coconut oil. Coconut oil occupies a unique position in canine brain health because of a specific mechanism particularly relevant for older dogs. It involves medium-chain triglycerides, a fat found in high concentration in coconut oil, and their relationship to brain energy metabolism. The brain's primary fuel source is glucose. In a healthy young brain, glucose metabolism functions efficiently and the brain receives an adequate and continuous energy supply. As dogs age, and particularly in dogs developing cognitive dysfunction syndrome, something changes in the brain's glucose metabolism. The efficiency of glucose uptake and utilization in neural tissue declines. Neurons receive less energy than they require. Cognitive function suffers as a direct result of this energy deficit. Medium-chain triglycerides provide an alternative fuel source for the brain. When metabolized in the liver, MCTs produce ketones, energy molecules that the brain can use directly and efficiently, independently of glucose metabolism. Research has shown that for an aging brain whose glucose metabolism has become less efficient, MCT-derived ketones represent a meaningful supplementary energy source that can partially compensate for the glucose metabolism deficit. A study published in the British Journal of Nutrition found that dogs fed MCT-supplemented diets showed significant improvements on cognitive assessment tests, including better performance on spatial memory tasks, improved learning speed, and reduced cognitive error rates compared to controlled dogs fed standard diets. The effect was most pronounced in dogs with existing cognitive dysfunction, precisely the animals for whom the alternative energy pathway is most needed. 1/4 to 1/2 teaspoon of virgin coconut oil per 10 pounds of body weight, added to food once daily, is consistent with the quantities used in research protocols. It should be introduced gradually to avoid digestive upset, and quantities should be kept conservative. Coconut oil is calorie-dense and can contribute to weight gain if used in excessive amounts. 5 foods: blueberries that cross the blood-brain barrier and protect neurons from the inside, fatty fish that build the physical structure of brain cells, eggs that supply the raw material for memory neurotransmitters, pumpkin that feeds the gut bacteria that feed the brain, and coconut oil that offers aging neurons a second source of fuel when the first begins to run low. None of these require a prescription, a specialist, or a significant change to your dog's daily routine. They require only the understanding that the brain sitting inside your dog's skull is an organ, a remarkable, complex, continuously working organ, and that what you feed it determines, in measurable and documented ways, how well it functions, how long it stays sharp, and how fully your dog gets to be present for every moment of the life you share. Always consult your veterinarian before making significant changes to your dog's diet, particularly if your dog has any existing health conditions or is on medication, as some foods can interact with specific conditions or treatments. If you are already feeding any of these to your dog, or if you are going to start, drop a comment and tell me which one. I read every single one. Like this video if it gave you something practical to use this week, and subscribe if you want to keep going deeper into the science of keeping your dog healthy, sharp, and fully present for as long as possible.